So today we'll be talking a little bit about the Clean Plant Program at FPS, Foundation Plant Services at UC Davis. And I'll go over just the mission. If you guys, some of you, I'm sure some of you are familiar with FPS, but if for the people who are not familiar, I'll go quickly about our mission. So basically, we produce clean plant material and we maintain it and test it. So this is kind of an important mission because starting clean is an important step of the vineyard management. Also, we are an importation center. So if you want someone you want to get material from France, Italy, international sources, it has to come through us because we do a lot of tests, and I'll go over that. So that's one of the services. Also, we coordinate the release of the new plant material release from the breeders. And you see me here talking to growers, to scientists. We do a lot of outreach. Why grapevine viruses are important? So grapevine hosts about a lot of viruses, and it's the most infected crop in the world. This number in red, because last year it was 70. This year, it's 75 known viruses are known to be reported in grapevine. Fortunately, not all those viruses are causing problems. I'll go over quickly some of the main groups. So we'll talk about Nepo viruses, rugoswood viruses, and leaf rolls, and some of the new emerging viruses like red blotch and grapevine pin grease. Quickly here, Nepo viruses comes from nematode transmitted polyhedral viruses. You see several of them that it's reported in grapevine. Not all of these viruses are present in the United States, but we test for them because they are reported in Europe. The ones that are highlighted are the ones that we found here, plus others. Some of the symptoms, here is the yellowing, and this is why it's concern, because it hits our pockets, it hits the crop, so this means we need to take care of it. You see healthy versus infected. The second group is the rugoswood, and this group of viruses cause the damage on the cylinder or the wood of the plant. So this means that you don't see a foliage symptoms for this group. We have to peel the bark to see the symptoms. You see the difference between the infected and the healthy. And the group under this disease group is you have Rhopistris stem pitting, copper stem grooving, LN33, and the corky bark. Again, this number of viruses is growing too. You see, up to last year, we have grapevine virus A to F. This year, we reported, there was a report for two additional, one from New Zealand and another one from France. And we just encountered a novel one here in California. When I say here at FPS, this means that maybe the virus is still in the pipeline, so it doesn't mean that it's spread. So this is still under study. Again, the symptoms. Sometimes it's going to be really show nasty symptoms. This is the copper 5BB. And here you can see the corky bark symptoms. I'm sure everybody is familiar with leaf roll disease. It's the most world known disease in grapevine, and it has a lot of economic impact, up to 40% reduction of the yield. And it also graft transmissible. It can be transmitted by mealybugs. Why? How it looks, the symptoms. If this is leaf roll 3 here, and it's kind of, you see some reddening. It's very similar. There is a high similarity between the symptoms induced by red blotch and leaf rolls. And this was one of the reasons that red blotch went undetected for a long time, because also we don't have the detection methods for it. Again, sometimes in white varieties, because there is no reddening in the leaves, you see yellowing and down rolling of the leaves. Again, it's important because of the effect on the yield, and here you can see healthy versus infected. This is a little bit more details. So we say leaf roll disease, but there is several species of viruses under the leaf roll disease, and they are different taxonomically. Why this is important for us to know? Because methods of transmission, the means of transmission are different between those viruses. Here we are talking about the new emerging diseases, the red blotch. Mark and Sudi gave a talk yesterday, and this is one of the viruses that he encountered. Another new disease is also the Grapevine Pinot Gris disease. This is, if you talk about the epidemiology of the spread of red blotch in Europe, in United States, you can compare the Pinot Gris virus like the red blotch of the Europe, because so far there is no report for red blotch from Europe, but this virus is widely reported there, which is the Pinot Gris virus. So the symptoms start to be noticed in the northern Italy and Trentino region. Using the high-throughput sequencing, which I will be talking in a minute about, they discovered this new virus that's showing some symptoms. And this virus was recently also reported here in Napa. So this is just a quick, outline about the viruses and the diseases. So what we can do to manage these viruses and diseases. So today I will focus on the first point, plant disease, which is a clean plant material, which is the title of our session. You can also do virus control of the vectors or develop resistant plant material. So for grapevine or other crops, we work closely with California Department of Agriculture, with the CDFA, and there is a certification program. It's a voluntary program, but most of the nurseries are basically now involved in the certification. So far at FBS, we have grapevine certification for grapevine, fruit trees, and strawberries. And this just to show you, this slide is just to show you the important impact, some economic studies to show the impact of a clean plant material. So for California, there is a study that in the northern San Joaquin Valley, there is about a benefit of using plant material, and it ranges 29 to 226 per hectare. The most finding one also, there is another study from New York, but just for one virus here in North Coast, which is grapevine leaf roll 3, the benefit of using the plant material is more than $50 million. This just tells you if a program like Foundation Plant Services or other places can cost up to $5–6 million and produce clean plant material, this case, we have only one virus, and you see what's the benefit of, or I will say the damage of not using clean plant material. Again, it's also important to make sure that we don't introduce a new pathogen, new diseases. Here you see the xylella and the plum pox in case of fruit trees. So we want to make sure also we will rely on you guys to spread the awareness that it's very dangerous to smuggle plant material. So this is when this happens, you think about it like it's maybe not affecting neighbors, it will not affect the industry, but sometimes can leave big impact. I will give you an example. In case of plum pox disease, which is a disease in fruit trees, the cost of eradicating this disease from Pennsylvania is more than $100 million. And it's still work in progress in Canada, which now they give up, they cannot eradicate it. In New York, Mark Fuchs and other people were heavily involved in a lot of effort to eradicate this virus. We don't know how it came, but it's important to spread the awareness about the importance to use legal channels to introduce plant material to the country. This is where we serve as a national introduction center, basically. We work closely with APHIS, which is basically USDA Department of Animal Plant Health Inspection Services. So we carry permits that we can introduce the plant material, run the test, and release it to the grower. And then sometimes, I just want to mention this, sometimes if you look for a plant material, check the database here in the state and make sure that you are not introducing a material already that's available for you to use. To do this big job, you need to work as a network, and luckily, since 2008, we had something called a National Clean Plant Network, and within this National Clean Plant Network, different centers are coordinating efforts and working together to make sure the support is available, the communication is available for such important effort. And this is a federal fund, so this is not, I can say it's the main goal of this fund is not to do research to discover new things, just to maintain the plant material, to clean the plant material and make sure you don't miss new plant material. Main crops in National Clean Plant Network, and these are the centers, different centers in the country. So we come to our slogan or to our model. This is from FPS here. We produce the material, we clean it if it's infected, and we sell it to the nursery. They grow it in their increased block. The nursery will sell it to the growers. So the slogan is, if you start clean, hopefully you will stay clean. And this is how we do it. If we receive a plant material from an imported plant material, local plant material, or a foreign selection, we start with doing the testing. And with the testing, if it's the plant material, I will go over the testing methods that we use. If the plant material is infected, we do something we call tissue culture to eliminate the virus. Then the plant material now, it's become, if all tests are negative, it's now generation 1, we put it in the foundation. We have to do some additional tests to make sure that the plant is true to type. Then we register it, and it will be released to the nursery to get to plant in their increased block. So what happens here, this can take up to 2 to 6 years, depends on the status of the plant material. If the plant material is infected, it will take longer to release it because it has viruses. What are the tests that we use? I'll go quickly over those different type of tests, and as you see, we combine different methods, and this, we realized that by doing this, we had good result, even with the viruses that we don't know, and we are just novel, and I'll show the case of red blotch. We found that combining different test methods is very important in this case. So quickly, we do biological indexing, which is biology using plant material here as an indicators. We do some serology, what we called ELISA, molecular PCR, and the new technique is high-throughput sequencing. Now, with the test required, these are the tests that we do with molecular and serological. This is what I'm sure you guys heard about Protocol 2010, material by nurseries. This is a list of viruses that we test for this protocol. Not all those viruses are here in the state, but because the source of the material mostly from Europe, we want to cover our bases just to make sure to collect some of these material. Now, the problem with these techniques that you need to know the pathogen to be able to develop an assay for it. So and sometimes it can miss some of the strains or the types of those viruses. So we need to move to a broader technique. In this case, the broader technique, and this is where there is some small herbaceous plants. If the virus can be mechanically transmissible for those plants, it will show some symptoms, but it doesn't work for all plants. So here you can see this is a cucumber, and if the virus we inoculate it within 7 to 21 days, if the virus is mechanically transmissible, you will see some symptoms. This is Nicotiana bentamiana, and this is the infected, and this is the healthy. The other type, this is the herbaceous. The other type of test we do is also called biological indexing, woody indexing. With the woody indexings, we use some woody plant in the field that they will show symptoms if the plant has a virus. How it works, we collect the plant material, we grow it, and we graft inoculate basically 2 buds from the source plant and monitor it for 2 years and look for the symptoms. And this is basically a group of diseases, and here I will emphasize in the word diseases, because this will show you only if the plant is showing symptoms, but you cannot go and look, what's the virus. So you need to do further work to identify the virus. But here a list of the most common indicator. We have the, St. George can show some viruses if it's infected symptoms. LN33, red blotch, and leaf roll is a Cab Franc indicator. We can show nice symptoms, and this is what you see here. And the last one is copper 5-VP, which is copper stem grooving. The advantage of this, I don't need to know the pathogen to be able to develop an assay. If the pathogen is there and it's reported to show symptoms, you will see the symptoms. But again, the disadvantages of this biological techniques, that a lot of those diseases can show no symptoms, can miss basically you have low titer. Some viruses will not show symptoms. It's time consuming, take 2 to 3 years to be, to be finished, and it depends in a lot of other things like the weather. If some viruses, if you wait, if you get a week of hot temperature, the symptoms will disappear. Just to give you an example. Also the titer of the virus, the type of the virus, if the successfully graft that virus and moved to the weather indicator. We had several cases where we did all the known assays and still we don't have identified a pathogen, and this is where this new technology comes handy. It's called high-throughput sequencing, next-generation sequencing, genomic sequencing, some people will call it, and it's rapid, accurate, and efficient, and it will give you a full, complete picture about the pathogen in these plants. So we say it's a comprehensive picture of the entire microbial profile of the sample. Just you generate millions and millions of reads and sequences, small sequences, and this is how it works. Just quickly, you have the disease plant, you do some lab work to extract the total nucleic acid from the plant material, you send it to sequencing platform instruments, it will generate tons of genomic data. This genomic data, you take it with the computer, the software, you compare it to a reference gene bank where you have also people will deposit information about pathogens. If the virus is there, if the bacteria is there, it will tell you, yes, this plant has bacteria or a virus or a fungi. So this is, in this case, you see that we have reads or sequences for viruses and viroids. This technique is evolving and it's growing quick, very fascinating things. Soon, we hope that you will be able to do this in the back of a truck. And if you see here, this is a small sequencer where you can put the plant material and get the sequences. And they tried it on the recent Zika outbreak. You see, this is what happened in Brazil, that they use this sequencer to characterize the Zika strain of the virus, and we call it lab in a wheel. Why we use it? We use it to study diseases of unknown. We used it to discover the red blotch. We use it for discovering the causal agent of the Pinot Gris virus disease, resequencing of known viruses. You have some countries also are using it at the border to make sure there is no new pathogen comes to the country. So we talked about biological indexing, and biological indexing is a requirement for certification. It's one of the requirements that we cannot release the material before doing biological indexing, which takes up to 2 to 6 years. So with this new technology, we want to see, can high-throughput sequencing replace HTS, replace biological indexing? So this is where we need, when you talk to regulators, they don't listen if they don't see proofs and published data. So this is where we did a side-by-side comparison, and we used the common grapevine viruses, including red blotch and Xylella, which is a bacteria. And preliminary finding, we found that it's much more superior in detection and can detect all the pathogen on target. Not only that, this is just a quick calculation of the cost of the conventional methods. It's cost about $1,900 to test a selection, and it takes 2 to 3 years. With this high throughput sequencing, HTS, now the cost about $500, and it takes 1 to 2 months. The most important advantage that it will detect all known and unknown viruses or strains. When we start using this technique in 2007, it cost about $5,000 to produce 6 to 8 million reads. Now it costs much, much less, and the price is dropping. We published these data, so based on this data, after we publish it in the journal Phytopathology, we start talking to the APHIS people, the USDA people, and they said the regulatory, yes, we have now some evidence that this technique can be used to accelerate the release of the plant material. So now we have the advantage of this provisional release that the growers, the nursery, they will start propagating the plant material earlier, 2 years earlier than the final release. So what we called provisional release. So I can give the plant material base of the negative with there's no viruses detected by high throughput sequencing, but we still want to do the long term. But when the grower has the access of this plant material, now they can start propagating this plant material they are not allowed to sell it, but when we give them the final release, they can be basically gain a lot of time, and time means money here. So again, this is basically has a lot of impact. So there is a coordination between countries, and still it's in work in progress. Again, it's a very important technique. It's a handy technique, but it has some technical challenges. And also, I would like to focus on the establishment of the biological significance of these viruses. Because this is a super, super sensitive technique, it can detect things that we don't know if had any effect in the plant. We are in the process of developing techniques, but I want to focus on this because of the short of the time. So if we know, if we find a pathogen, a virus, a sequence in a plant, doesn't mean that this virus will cause an effect or cause the disease. So we need to do further like that. And we need to focus only in the high economic impact regulated viruses. And this is what you see here. No decision should we take before we do 4 steps. We need to do graft transmission to make sure that this virus is graft transmissible. We want to make full Cox postulate. When you take the pathogen and you try to introduce the symptoms again, and when it's widely spread, and also when you assess the symptomic or economic impact. A long list of viruses discovered by high-throughput sequencing, but I will give the red blotch as an example. We did confirm that this is a virus that it's graft transmissible. It's widely spread. At Cornell, they fulfill Cox postulate, and it's impacted the industry, even considered as a threat for the industry. Only based on all those steps, now we consider that red blotch is added to the certification in California, New York, Oregon, and Washington. But is red blotch a new disease? It's not. Because if we went to the herbarium and we tested some plant in the herbarium, we were able to find this disease in the herbarium specimen, a cultivar called Early Burgundy. The plant material was collected in 1940. So the technique will discover new viruses, but the viruses will be just because we have the knowledge how to do it now. Okay, so to summarize, we know that HTS is very sensitive and comprehensive measure. We are hoping that within a few years, it's going to be replacing the biological indexing. So what if this plant came to be infected? Is this the end of the world? No, there is something called Meristem culture, where we take the plant material and we can clean it from the virus. And the principle behind that, this is a Meristem, where you cut this piece here, and this is the virus. The plant material, basically, the plant cells divides faster than the virus. So if you take this part from the plant, there is a high chance that you started with a new plant material which is free of the virus. It's not always guaranteed to work because some viruses are more stubborn, but there is a high chance that some viruses can be eliminated like this. If you see, this is a small meristem excised under the microscope. Within 5 to 12 months, you will have a clean plant if it's successful meristeming. We have to repeat the test, all the tests that we did after, basically, if we find this plant tested with red blotch, we have to test it after we finish the meristem tip culture to make sure that it's eliminated. We do also DNA verification to make sure that it's true to type, and different methods of DNA identification. This is our vineyards. The last thing I will cover, a couple of just minutes, is basically, this is what we sell. We sell plant material, disease-tested, professionally identified, true to variety, and this is the price. But I hope that I convince you that we are doing a good job. But I will show you some data that also support that. What you see here is what's the long-term benefit of such a clean plant program. Let's take Russell Ranch plant material or classical plant material. So when we found red blotch and Pinot Gris, we went back and we tested our vineyard. So just to make sure what's the percentage. So in Russell Ranch, because everything in Russell Ranch passed through shoot tip culture, and we tested for the protocol 2010, there was no red blotch or Pinot Gris at that time. For the classical, because when you do meristemic shoot tip culture, you eliminate the viruses that you know about and the viruses that you don't know about, based on the principle. With the classical, out of 4,600 vines, almost only 15 vines tested positive. This means that the cleanup, the system, if you remember at the beginning, I said it's a good approach to use different methods for detection. Also, up to 20 to 30 million grapevines tested per year trace back to FPS. So if you start with a clean plant material, this gives you a good start. Again, I presented this high benefit impact. And just the last few years, a postdoc did a study where they tested, Carrie Arnold, she did basically sample plant material based on the age. So in 1800s, 1900s, and 2000, and what we noticed that the new plant material that the nursery and the growers now looking and have more clean plant material. So there is less virus, except for the case of red blotch, because that was not, we don't have tool to test for that virus at that time. Some acknowledgement for the people that supported us. And there was a question yesterday about that sampling and cross-contamination. This is the link. This is if you go to Foundation Plant Services, under virus diagnostic, you will see some directions, a video, and some good directions for how to collect plant material. Thank you very much.
Thank you.